If I told you that the cutting-edge regenerative solution for severe perimenopausal symptoms involved injecting highly purified DNA from male salmon trout directly into the
I
labia.
mean, you would probably think I was describing some bizarre internet rumor, right?
Exactly, or like some experimental biohack that just went completely wrong.
It sounds completely absurd at first glance. I mean, we are talking about taking genetic material from a marine species and literally using it to reverse structural tissue collapse in
Wow.
human reproductive organs.
It's wild.
It is. But the clinical data behind this is fundamentally rewriting how regenerative medicine approaches genitourinary syndrome of menopause, or GSM.
Right. And so our mission today for this deep dive is to look at the hard science behind these intimate polynucleotide injections.
Yeah, we've got a lot to unpack.
We really do. We are pulling from a fascinating stack of sources today. We've got the Palmieri exploratory studies on vulvovaginal biorevitalization, some real-world aesthetic clinic protocols, and of course, the latest molecular research on tissue regeneration.
Which is moving so fast right now.
It really is. And the reality is, up to 84% of postmenopausal women experience these intimate structural changes.
Which is just a massive number.
84%, yeah.
Wow.
Yet so many suffer in complete silence, just assuming that the profound loss of tissue elasticity and, well, the resulting pain is just an unavoidable toll of aging.
That is just something you have to put up with.
Exactly. So today we are going to explore how science is moving past those temporary fixes and actually regenerating this really delicate tissue at a cellular level. Okay, so let's unpack this.
Yeah, so to really grasp the magnitude of what these polynucleotides are doing, we have to first look at the sheer violence of the hormonal cliff
Right.
that triggers vulvovaginal atrophy in the first place.
Where's the cliff?
Because during a woman's reproductive years, estradiol levels are incredibly dynamic. I mean,
Yeah.
they surge and fall with the cycle, sometimes peaking around 800 picograms per milliliter.
800, okay.
Yeah, and this estradiol is basically the primary signal keeping the whole vulvovaginal architecture robust. The estrogen receptors in the tissue are constantly being stimulated.
So they're constantly telling the body to, like, maintain blood flow, produce collagen, keep things hydrated.
Exactly. But then, perimenopause hits and that reservoir just drains. Those estradiol levels plummet from around 800 to consistently below 30 picograms per milliliter.
Wow, from 800 to below 30.
Yeah, the signaling network just entirely shuts down.
So what happens physically when that signal drops off so aggressively?
Well, the physical manifestation of that shutdown is a really rapid structural collapse. It starts with the labia majora losing its adiposity.
Like the fat pad.
Right, the fat pad literally diminishes, resulting in this visible loss of volume. And then beneath the mucosal surface, the subepithelial connective tissues become entirely depleted of their collagen and elastin networks.
So the whole structure just sort of gives way.
Yeah. And the mucosal rugae, those accordion-like folds that allow the tissue to comfortably expand and stretch,
Right.
those completely flatten out. And at the same time, the local vascular density decreases, meaning basically less oxygen and fewer nutrients are actually reaching the area.
I mean, the analogy I keep thinking of from the sources is it's like a mattress, right? Losing its internal springs and the stuffing all at once.
That's a great way to look at it. It's not just a surface issue. The entire scaffolding is giving way.
Right. So you're left with a microenvironment that is incredibly thin, fragile, chronically dry, and just super prone to micro tears.
Exactly. The tissue just loses its mechanical integrity. So when that extracellular matrix breaks down, just the friction of normal daily movement, let alone intimacy, it triggers this chronic localized inflammation.
Okay, wait, hold on a second. If the primary immediate symptom is friction and dryness and irritation, why not just, go to the pharmacy?
I get that question a lot.
Right, because there are entire aisles dedicated to over-the-counter moisturizers and lubricants. I mean, why go through an invasive medical intervention for something a topical cream seems perfectly designed to handle?
Well, so over-the-counter lubricants absolutely serve a purpose, right? For immediate surface friction, they're great, but they are essentially cosmetic bandages over a structural failure.
Like putting lotion on the broken mattress.
Exactly. They don't interact with the extracellular matrix at all. They cannot trigger the synthesis of new collagen or elastin fibers, and they definitely do not signal the local blood vessels to regenerate.
They just sort of sit there.
Right, they sit passively on the epithelium. So to actually reverse the atrophy, we need an intervention that penetrates the tissue and communicates directly with those dormant cells.
We need something that actually rewrites the biological instructions.
Precisely.
Which brings us back to the star of today's stack of clinical studies, polynucleotides, or PNs.
The PNs, yeah.
So the sources detail the use of polymers extracted specifically from the gonads of male salmon trout.
Which is the part everyone gets stuck on.
Oh, absolutely. My immediate question when reading this was, what about the immune system? How does the human body not instantly flag fish DNA as a massive foreign invader and trigger some severe autoimmune response?
It's a totally fair question. And it forces us to kind of at least reconsider how our immune system actually recognizes threats. Because pathogens aren't typically identified by their raw nucleic acids.
Oh, really?
No, the immune system is usually looking for specific proteins, peptides, and surface lipids, like the ID tags or the packaging around the DNA.
Okay, so if you strip away the packaging, the alarm bells don't ring.
Exactly. And that is the function of the PNHPT process that's highlighted in the sources.
High purification technology.
Right. It's this rigorous, multi-step, really high-temperature extraction process. And it basically obliterates all the proteins, all the lipids, and any potential immune-triggering elements,
Leaving just the raw DNA.
leaving highly purified, biologically active nucleotide building blocks. Because those fundamental molecular letters, adenine, cytosine, guanine, thymine.
The ACGT from biology class.
Exactly. Those are universally recognized across all species. So the human body doesn't see a salmon cell, it just sees a reservoir of raw, high-grade biological building materials.
Wow. So the structural homology is close enough that the human tissue just accepts it natively.
Natively, yeah.
Without a fight. But these polynucleotides aren't just like inert building blocks waiting around to be used, right? The literature describes them as highly active signaling molecules.
Yeah, and what's fascinating here is how active they really are. When they're injected into the atrophic tissue, they immediately begin to alter the local microenvironment.
How so?
First, they physically interact with the local fibroblasts. Specifically, they bind to what we call the A2A receptors on the surface of these cells.
Okay, the A2A receptors.
Right. And this binding action effectively wakes up the dormant fibroblasts and aggressively upregulates their production of type I collagen and elastin.
So they basically act as site managers, right?
Right.
Just commanding the factory to reopen and start producing the structural scaffolding that the estrogen drop shut down in the first place.
That's a perfect analogy. But they actually do more than just issue commands. Through a process of passive hydrolysis, these polynucleotide chains slowly and progressively break down in the tissue over a period of weeks.
Oh, so they don't stay intact forever.
No, they degrade. And as they degrade, they release individual nitrogen bases and nucleosides directly into the extracellular matrix.
Wait, so they are handing the fibroblasts the blueprints via those A2A receptors and then simultaneously breaking themselves down to provide the raw materials needed to actually execute the blueprints.
Exactly that. It's an elegant system. And meanwhile, before they fully degrade, these long-chain polymers organize themselves into a complex 3D hydrogel scaffold right within the tissue space.
A hydrogel.
Yeah, this structural web has a massive molecular weight and is incredibly hydrophilic, meaning it binds and traps substantial amounts of water, which provides immediate, really deep structural hydration.
Which is exactly what that dry, atrophic tissue needs.
Right. And as a bonus, this hydrogel also acts as a free radical scavenger, neutralizing the reactive oxygen species that drive the chronic inflammation we see with VVA.
Okay, so we have this massive multi-pronged approach here. We've got epigenetic signaling, raw material delivery, structural hydration, and inflammation reduction.
It's doing a lot of heavy lifting.
Yeah. So let's look at the actual clinical application of this, because the studies map out a very specific protocol for administering this treatment, and it's an in-office procedure, right, involving a series of intradermal injections.
Yes. The clinicians are essentially targeting the superficial lamina propria. That's the delicate connective tissue layer directly beneath the epithelium of the labia majora, the labia minora, and the vestibular area surrounding the introitus.
The introitus being the vaginal opening.
Correct. And to manage patient comfort, what is obviously a highly innervated sensitive area, the protocol utilizes a potent topical anesthetic cream and a highly specialized 30G needle.
So a tiny needle.
Very tiny. It minimizes the mechanical trauma during the delivery.
Now, the sources outline a really fascinating synergistic protocol here, because they don't just use the polynucleotides in isolation, do they?
No, they often pair them with hyaluronic acid.
Right, HA. The clinical framework refers to the PNs as the primer and the HA as the consolidator. And some of the newer regenerative formulations like Nugen, I think it's called.
Yes.
They physically combine the two molecules into a single syringe alongside mannitol. But, okay, I have to ask. If hyaluronic acid is already so effective at drawing in moisture, why couldn't a clinician just use standard HA dermal fillers down there?
Like they do for the face.
Yeah, exactly. People use HA all the time to restore volume in their lips or cheeks. Why do we need the complex DNA extraction at all if HA already plumps things up?
It's a great distinction to make because hyaluronic acid is unparalleled as a hydrating agent and volumizer. I mean, it immediately binds moisture and provides visible tissue plumpness, but HA is not an active cellular signaling molecule.
Ah,
It does not command fibroblasts to synthesize permanent collagen. It alters the fluid dynamics of the tissue, sure, but it doesn't change the underlying cellular behavior.
so it plumps the tissue, but it doesn't actually build a new scaffold.
Exactly.
Yeah.
The polynucleotides are the biological managers. They are the ones initiating the tissue regeneration. But they work best in perfect synergy with HA.
How does that synergy work?
Well, the profound hydration provided by the hyaluronic acid creates the ideal moisture-rich microenvironment that those fibroblasts desperately need to stay vital and metabolically active.
Oh, I see. So the HA floods the factory with the water and power required to keep the machinery running, while the PNs actually hand out the work orders and supply the raw materials.
That's exactly it. And that formulation you mentioned, Nugent, it includes mannitol for a very specific biochemical reason, too.
Right. I was wondering about the mannitol.
Yeah, mannitol is a powerful antioxidant that scavenges free radicals, but its primary role in this synergy is to inhibit hyaluronidase.
Which is what exactly?
It's the enzyme in our bodies that naturally breaks down hyaluronic acid.
Oh, so it stops the HA from disappearing too fast.
Exactly. By protecting the HA from rapid degradation, the mannitol ensures that the optimal hydrated microenvironment is maintained long enough for the polynucleotides to actually complete their slow-release regenerative work.
Okay, so we've mapped out the cellular architecture, we understand the biological synergy, but I think the real measure of this intervention is the human impact, right?
Without a doubt.
We are moving from the molecular mechanisms to the actual clinical outcomes for these women, and the exploratory studies led by Palmieri yield some really striking data.
The reduction in clinical symptoms after just a relatively short cycle of these localized injections is, frankly, profound.
I mean, we are looking at conditions that severely degrade a woman's quality of life. And following the treatment cycle, self-reported itching and soreness dropped by over 50%.
50% is huge for chronic pain.
And even more significantly, dyspareunia, which is painful sexual intercourse, and severe vaginal dryness, those plummeted by nearly 60%.
Those statistics represent a fundamental restoration of daily comfort and intimate function. And it's achieved through a minimally invasive ambulatory procedure.
Right, you just walk in and walk out. Now, the stack of research also includes one study that I found incredibly compelling because it bridges the biological outcomes with the psychological data.
The pair skill study.
Yes. Researchers took 47 menopausal women undergoing this treatment and evaluated them using the pair scale.
Right, the personal assessment of intimacy in relationships. It is a highly rigorous, validated psychometric tool. It's basically designed to separate the different dimensions of a couple's connection.
Because intimacy isn't just one thing.
Exactly.
So the pair scale relies on 36 distinct items, and 30 of those items probe the non-sexual, day-to-day, emotional, and intellectual intimacy of the relationship, like how the couple communicates, their emotional closeness, their shared activities.
And what did the data show there?
Well, the study showed that after the polynucleotide treatments, those 30 non-sexual items showed zero statistical change.
Which makes perfect sense, right?
Yeah.
I mean, an intradermal injection isn't going to suddenly resolve underlying communication issues or, shift the emotional baseline of a marriage.
Exactly. It's not marriage counseling in a syringe.
Right.
But the remaining 6 items on the pair scale, they specifically target sexual satisfaction and physical intimacy. And out of those 6, 5 demonstrated highly significant improvement.
That divergence in the data is just critical for validating the intervention because it essentially isolates the physical mechanics of intimacy from the broader emotional landscape of the relationship.
It proves it wasn't just a placebo effect.
Right. The treatment didn't induce some generalized placebo effect of well-being. It solved a localized biological failure, which then directly translated to a quantifiable improvement in physical intimacy.
And here's where it gets really interesting. As we were analyzing the methodology of these studies, a massive realization emerged regarding how these results were actually achieved.
This is perhaps the most important takeaway.
Yeah. These profound clinical benefits, the 60% drop in dyspareunia, the restored sexual function, they were realized without the clinician ever needing to treat the deep internal vaginal canal.
The injections were entirely confined to the outer vulvar structures.
Which is huge because for anyone unfamiliar with the reality of severe vulvovaginal atrophy, the mere introduction of a speculum for an internal examination can cause excruciating pain.
It can be traumatizing.
So the idea of undergoing internal energy-based therapies like, fractional CO2 lasers, that's a massive psychological and physical barrier for a lot of women seeking help.
Absolutely. And by avoiding the internal mucosa entirely, this protocol fundamentally shifts how we view the biomechanics of dyspareunia. You really have to consider the anatomy of the introitus, the vaginal opening.
What's happening there mechanically?
Well, the vestibule is incredibly dense with nerve endings. So when the subepithelial connective tissue here loses its elasticity and the rugae flatten out, the opening itself becomes completely rigid.
It essentially loses its ability to safely expand.
Right. So when you attempt penetration through a rigid atrophic introitus, you are mechanically stretching fragile tissue way past its breaking point.
Ouch.
Yeah. This immediately fires the nociceptors, which are the pain receptors, triggering localized inflammation, micro-tearing, and often causing the surrounding pelvic floor muscles to defensively spasm.
Wow.
So the pain radiates internally, but the primary bottleneck, the mechanical trigger, is right there at the entrance.
So by utilizing polynucleotides to selectively rejuvenate the outer ring, restoring the rich moisture, rebuilding the elastin fibers, and regenerating that superficial lamina propria, the introitus regains its functional elasticity.
Exactly. It can accommodate expansion again. You remove the mechanical friction point, which halts the firing of those pain receptors in the first place and prevents the cascading pelvic floor spasm.
So you are effectively healing the internal experience of intimacy just by optimizing the structural integrity of the external entrance.
That's the beauty of it. It completely reframes the approach to treatment.
It really does. So what does this all mean? Bringing this all home to you, the listener. If you, or someone you care about, is navigating the complexities of perimenopause, it is vital to understand that the loss of intimate structural integrity is not a permanent life sentence.
It's really not.
It is not an inevitable decline that you just have to silently endure. The frontiers of regenerative medicine are rapidly moving beyond aesthetic dermatology. The same advanced cellular signaling that, rebuilds collagen in the face is now being deployed to restore dignity, daily comfort, and intimate function.
We are truly witnessing a transition from merely masking the symptoms of aging with temporary lubricants to actively reprogramming the atrophic tissue to rebuild itself.
Science is delivering non-hormonal regenerative interventions that can actually replace the extracellular scaffolding that time and estrogen deprivation strip away.
And if we connect this to the bigger picture, it forces us to consider the broader implications of this technology. I mean, think about it. We're taking highly purified DNA fragments from marine species, injecting them into localized estrogen-deprived human tissue, and successfully commanding that tissue to essentially reverse its biological clock.
It's incredible.
The polynucleotides essentially hack the local cellular microenvironment, forcing the fibroblasts to ignore the systemic hormone deficiency.
They just bypass the need for circulating estrogen entirely.
Right. So if localized epigenetic DNA signaling can successfully reconstruct the architecture of reproductive tissue without altering the patient's systemic hormone profile, what are the limits here?
Oh, wow. Yeah.
Could this approach eventually provide an alternative to systemic hormone replacement therapy for other aging organs in the body? Are we looking at the foundation of a whole new medical paradigm where we treat the structural decay of aging organ by organ using targeted nucleotide blueprints?
It is a profoundly provocative concept to think about, and regenerative medicine is clearly just scratching the surface of what's really possible with cellular signaling. Thank you for joining us on this deep dive. Keep exploring, keep questioning the consensus, and we will catch you next time.